Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for the replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions in the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Nigel Shipston from FUJIFILM Diosynth Biotechnologies.
So the title of my talk today is Contract Development and Manufacturing Organization's Perspective on the Development of Bioprocesses for Clinical Manufacturing Success. I'm Nigel Shipston. I'm a Director of Program Design at FUJIFILM Diosynth. And my role at the company is to work very closely at the early stages of scoping a program to define the appropriate scope. And one of the key outputs from our team and myself are proposals that are aligned as closely as possible to client needs. So I'm just going to move to the first slide. The slide pack is a mixture of non-technical and a few technical slides. I'm going to start off with a gentle introduction, just overviewing the presentation. So we're going to cover general aspects of working with a CDMO, a contract development and manufacturing organization. And then we're going to touch on some of the goals and key features of process development and analytical development, particularly in the context of early phase development, so development of drug substance for early clinical phase studies. And finally, we're going to wrap that up with a brief summary of the points we've covered. So many of you perhaps are involved or expect to be involved in the selection process for a contract development organization, contract manufacturing organization. So I thought it'd be useful just to briefly touch on the main aspects of the selection process that many of you are perhaps already familiar with or will need to become familiar with. So really, it typically starts out with a request being made through whatever channels exist, a request for a proposal, otherwise known as an RFP, as we get into terminology. And then the CDMO will respond to that proposal request with a proposal, which is essentially a quotation with some level of detail around the scope involved. So the next step would be an assessment of the proposals received, and that may be carried out entirely by a client, a person as a customer, or it may involve one or more consultants working in conjunction with the client. Ultimately, the initial scoping will boil down to a short list. That may be 2 or 3 CDMOs who've made the first cut in the selection process. And typically that will be followed up with more detailed discussions and almost certainly a site visit. Following that, one of the critical steps would be to indicate some kind of commitment to the CDMO to progress beyond the initial assessment phase into contractual commitment. That's an important step from a CDMO's perspective because that initial commitment gets you more visible on their radar in terms of planning for potentially a project to land. So it starts to trigger the mechanisms for a CDMO to ensure that the capacity and the resources are available to undertake the work. And then underpinning the contract itself are 3 important documents: the statement of work, the master services agreement, and the quality agreement. And you can see the acronyms there, commonly used terms in our business. So then selecting a CDMO, many of you perhaps involved in the selection process, and the criteria really, depend sometimes on the individual needs, your individual needs, But almost always, right at the top of the list are going to be, has the CDMO got the technical expertise and the capability and experience demonstrated to deliver the work? And then additional important considerations are, does it have the capacity? Does it have the capacity at the right time? Does it have, as you get into more detail, the equipment, the appropriate equipment, and the facilities that you're looking for? Another consideration, of course, is cost or price of the program. It's usually not top of the list, but it's usually very high in the list of priorities. And then there are secondary considerations such as the location of the CDMO, the time zone, softer skills such as a willingness, a demonstrated willingness to be flexible and take a flexible approach to your needs. And underpinning this all is it's important to get a sense for, because people and relationships matter so much in this business, does the dynamic feel right? And I think, both from the CDMO's point of view and from the client's point of view, it's usually possible from early discussions and face-to-face meetings to get a real sense as to whether the relationship will work. And that is absolutely critical to delivery. So in terms of working with a CDMO, I think, the needs of the CDMO and the needs of the client, the business objectives are often different. They're different businesses, essentially. But for a program to work, those differences need to be aligned, and that's one of the key priorities to make success. So underpinning all the work that's to be undertaken is a legally binding document called a statement of work, and that governs the tasks that have been agreed to be performed. It's usually tied to the contractual documents such as the master service agreement. And, unlike the proposal, it is legally binding. So the quotation, the proposal is a quotation, the statement of work is a legally binding document. But always remember that in early phase clinical development, the work is likely to be developmental in nature, and biological systems and molecules can be unpredictable. So it's a good idea to go into a program in early phase development with your eyes open to expect the unexpected. And then also keep in mind that CDMOs are very often they're dependent on third-party providers, suppliers of consumables, materials, ancillary services such as analytical services, characterization services. And so, they can sometimes be hostage to the fortunes and the priorities of those third-party providers. And again, this is where, a good CDMO will have already built a good relationship, person to person, with their third-party providers, as well as with the client. Very often, the project team is supported by a joint steering team, which is operates somewhat independently of the project team. And it exists very often to crunch key items that may come up in a project of a strategic nature, to keep the project team focused on the more immediate work to be delivered. So typical stages in the scope of work a typical program might look like this, as you can see on the screen. It would typically start out with a tech transfer phase where, depending on the nature of the program, it will either be a limited amount of information or an extensive amount of information. It depends really on what the starting point is. And it will likely involve at least some transfer of materials from the client to the CDMO. At the bare minimum, it could be the transfer of information, perhaps no more than a protein or gene sequence. And at the other end of the scale, it could be the transfer of a whole process with all the incumbent details of the process and associated analytical methods. So it really does vary. The next stages are typically process development, which could be, again, range from establishing a developed process, developing a small-scale model, right through to extensive development from scratch of a process. And in conjunction with that, developing at the same time a suite of analytical methods to support both the drug substance release and in-process testing. Almost always that process development stage will then be rounded off with what in the industry is often called a demonstration run or a process demonstration run, where the process is then finalized and locked prior to transfer to manufacturing. Typically, it's performed at an intermediate scale, and then that might be followed by subsequent engineering runs, which typically they might be more normally performed at the intended GMP scale, but they will be in a non-GMP context. So in other words, they might be using draft documentation rather than finalized documentation. And after that, typically engineering run or runs would be followed by as many GMP batches are needed to deliver the required clinical material. So choice of the expression system for your molecule, the system or the host, is very often one of the early critical decisions to be made when developing a strategy. And as you can see from this slide, the typical cell lines or hosts can vary according to the needs and the application. Really, the three main classes of host systems fall into either mammalian, insect cell, or microbial. And really, you know, in the context of a recombinant protein, there is quite a degree of flexibility in what system could be used depending on your molecule, but there will be most definitely criteria which might determine the precise system chosen. And this next slide just really touches on the kind of considerations that might be made. So, you know, the timeline for how long it takes to develop the appropriate host strain or cell line can become a critical factor. But overriding that will be considerations on the intrinsic protein or product properties. So, for instance, if glycosylation of the product is needed, that will likely influence the choice of the host system for expression. Other considerations are the product titer and whether the product can be secreted or not secreted, along with an overview and a general understanding of the likely cost of goods associated with expression system. Choice of expression system can also determine the purification strategies that are open to working with the molecule, and sometimes those purification processes can be very complex and very costly. The expression system may not be obvious, and in those cases, very often feasibility studies are often appropriate to make the right choices at an early stage. The important thing to bear in mind is that expression system can be with you for a very long time, and those decisions are very often made up front, so it's important to make the right choices at an early stage. So the goals of process development, particularly early phase, if we just look at the key aspects of process development, for early phase, they are to develop a process to produce drug substance and drug product that is essentially appropriate for toxicology studies, namely safety and efficacy in an animal model. And secondly, to be appropriate for the first in human or clinical phase 1 trials, usually to determine whether the drug is safe to be used in a human in efficacy studies. And there will be a prelude to determining safe dosage ranges. So the quantity and quality of the product needs to be sufficient, and also the scalability and the line of sight. The demonstration of that at an early stage is very useful to support future scale-up activities in the future. So key features of early phase development are aggressive timelines, usually because of commercial pressures. The process needs to be good enough to meet near-term needs in terms of purity and productivity. But remember, you're not looking to achieve perfect process perfection in those early stages. So usually it's a trade-off between product quality, productivity, robustness, and speed. So key challenges here are to expedite the initial stages of process invention and to avoid reinventing the wheel for every product. So if platforms are available, it's useful to be able to leverage off those as much as possible. And finally, the application of advanced analytics for product characterization. It's very important to determine as early as possible that you're making the right stuff from your expression system. So I'm just going to skip forward now to briefly the timelines on a program. One of the key questions might be, how long will that work take? And the answer is, it's probably going to be longer than you thought. So if we look at the timelines for a typical program of early phase process development, that typical timeline is going to be something of the order of 12 to 18 months. And that depends sometimes on whether the cell line or the strain and critical materials available, or whether they need to be developed as an ancillary program. Remember that the scope discussions and the agreement, that is often underestimated, and a typical kind of timeline to get from the proposal or the non-binding quote to a statement of work is of the order of 2 to 3 months. And then remember that further contract negotiations for master service agreements and the quality agreement typically can take 3 to 6 months. And on top of that are considerations that the client and the CDMO will need to align on, which are the program starting slot and the manufacturing slot. And these may be variables and moving parts during all these negotiations. So really, in summary to this very brief webinar, that the molecule and the asset, well, it's your baby. So we understand as a CDMO, your needs are very closely tied to the product, which is your baby. And choosing a CDMO partner wisely is a critical step along the way to contract process development, with the mutual goal being to build an effective long-term working relationship based on trust and transparency. Thank you for your time. And I'm happy to take any questions.
Great. So the first question is, if I'm not sure what expression system to use for my product, what would you advise?
So we briefly touched on that. And just to elaborate then, so I think the aspects that you need to consider would be the intrinsic properties of the molecule. So it's important to consider, for example, whether the molecule is glycosylated. And if it's glycosylated, the likelihood is that you're looking at a mammalian expression system, possibly an insect or yeast host might work, but invariably it's likely to be a mammalian system, and the most common expression system is going to be in Chinese hamster ovary cells. Other aspects to consider would be if there is any evidence of in the literature or in patents of successful expression of your molecule, if you have no internal information to guide that. And the third thing is really to consider an expression feasibility studies. So at FUJIFILM, we're very often used to conducting 2- or 3-way expression studies where we'll perhaps compare expression in a microbial system such as E. coli or yeast versus a mammalian system as CHO. And you can get, in those kind of studies, typical readouts from E. coli might be within the time frame of 6 to 8 weeks. In CHO, you're looking at closer to 10 to 12 weeks. And those studies can either be performed in sequentially, or in parallel.
Okay. And what are the critical purification challenges?
So critical purification challenges really are removal of product-related impurities and host protein, host DNA. So it's really, a combination of those aspects. So clearance of product-related impurities from your product is obviously important, as is clearance of host cell protein, host cell DNA. There may be other elements in there. For example, if the molecule is being expressed in E. coli, then endotoxin will be a consideration. So removal of endotoxin, which is a pyrogen, is critical. And again, ensuring that the bioburden is very low, approaching sterility for drug substance. So those are the challenges, and a balance against that is the fact that as you purify a molecule up, you're going to lose material. So really it's a case of balancing purification versus yield.
Okay, I think we have time for just one more question. If we didn't get to your question, we'll be sending those to Nigel and he can follow up with you directly. So go ahead and feel free to type in more questions and we'll just pass them directly to you and he can get back to you after the webcast. So the final question for today is, what are the biggest challenges in gene therapy you're seeing?
So I think that the biggest challenges in gene therapy right now, it's not a field of direct personal expertise, but the challenges are really scaling up processes. So a lot of the processes that are available are for, they're really operating at very small scale. And one of the real challenges is scaling up processes that are difficult to scale up, and therefore may need different approaches, different technological solutions to enable production of a molecule of the requisite quality. So this is one of the key challenges I believe in the industry, is really making, there are very few platforms out there, and those platforms that exist are difficult to scale up. And there's really one of the key challenges right now.
Okay, well, thank you, Nigel.
Okay, well, thank you very much, Leah, and thank you to everybody for your attention.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. As a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us for future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox in the new year. Bye.